Andy's lab studies the role of genetic diversity in herbivory and establishment and transmission of plant disease. So Andy's work in the true spirit of SID has always been highly collaborative, interdisciplinary, working at the intersection of ecology, evolution, genetics, development, plant pathology, and agriculture. So the fact that Andy can do a seminar tour and have 4 different seminar, departments or centers that he visits at Penn State is really indicative of that breadth. So Andy did his undergraduate degree at Miami University in Ohio, followed by his master's and PhD at the University of Michigan, before coming to Penn State as an assistant professor. Can I say what year?
Yeah.
Do the math. 1978. Woo. So over his time here since, he's mentored 7 postdocs and over 20 grad students, all of which, amazingly, have gone on to positions relating to science at universities, government, and private industry all around the world. So Andy has served in numerous leadership roles at Penn State, including Associate Dean for Research and Innovation, and most recently the Director for the Office of Innovation. So thank you, Andy, for your great science and for your service at Penn State. We're hoping you have exciting things planned for your retirement, and I think people are excited to hear about safe sex complaints.
Thank you very much. I've been boring people all across this university for the last 3 weeks with these just so stories. And the goal of these is really to tell interesting natural history stories about plants. Some of you may have read Rudyard Kipling's Just So stories, how the leopard got its spots, how the giraffe got its long neck, how the elephant got its trunk. Well, they're very Lamarckian, and there's no stories about plants. And so I've decided to do a sort of transect through like 46 years of experimental natural history. In my youth, I would look at the tremendous diversity of flowers, and I was interested in how the various traits, the positioning of the stigmas and styles, the positioning of the anthers, how the color, odor, shape, types of nectar rewards, and things of that sort, how these floral traits influenced pollen dissemination, the gathering of pollen onto stigmas, and so, for example, here we see anthers arrayed around the outside of the flower with the stigma on the inside. You don't get much pollen transfer within a flower that way. Other plants do it differently. There's differences in stigma heights and the ability to accept or reject self-pollen. But each of these plants has its own type of story. This is an interesting one. This has the stigma sticks out one way and the anthers the other way, and there's a polymorphism where some plants are left-handed, some are right-handed. Here you can see the left and right-handed. But anyway, I've been looking at these floral traits across, actually I haven't worked with very many species, but a few. But really, when I started this and throughout most of my career, when you looked at floral design or the entire floral display of a plant, the primary function of both is to promote mating success. And what I was interested in is how plants could get their pollen to as many stigmas as possible, and how they could gather as much pollen onto their stigmas, and who won this race from the stigmatic surface as these pollen tubes grew and achieved fertilization. So I was interested in, mating success, gathering pollen from lots of different individuals onto the stigma, gathering far more pollen than you needed, and the advantages of doing that. But that was in my youth. Now I'm a bitter old man. I was approaching an age where I may no longer be able to take yes for an answer. And so I've got a whole different perspective now. I'm interested in safe sex. Okay. So when pollinators, wind, rain, these bring microbes, including pathogens, to the flowers. For example, this bumblebee or carpenter bee, I can't tell from this close, it may have visited 150, 200 other plants before coming to the plant that I took the picture of. So this bumblebee may actually be bringing in pathogens and diseases from across the population, maybe across multiple populations, and bringing it to that flower. Moreover, nectar is a rich media for the growth of microbes. Nectar has, it's sugar water is what it is, basically. There might be a few other things in it. It's a rich growth for the media of microbes. The nectary also provides easy access to the vascular system, and hence pathogens can go through the nectary into the plant plumbing system and then be distributed throughout the plant from there. The stigma and style of the flower is also a rich media for the growth of pollen tubes. Remember, pollen tubes actually germinate, they grow. They're a little different than the sperm cells in animals, which basically just flap their way from one end of the tub to the other, like a rubber duck or something like that you wind up. That winds it up, sets it down, and flaps its way to the other end. But anyway, these actually grow, and the stylar tissue has the kinds of nutrients and energy compounds that make the growth, that stimulate the growth of these pollen tubes. Well, it's also good for microbes. The style also provides easy access to the ovules, and hence vertical transmission of diseases can occur that way for pathogens that come down through the style and get into the seeds. So, as you know, sexually transmitted disease, as you know, because of Giannis Antonovics, I know he's given at least 3 talks since I've been here about sexually transmitted diseases in plants. This is a system that he and his students have worked on, in which these Selenes, if they're infected with a fungus, they actually, instead of making pollen, they make fungal spores. So when a butterfly comes by, picks up these spores, then comes to a new plant and deposits them onto the stigma of the flower. Okay? And when you think about it in the terms of infectious disease dynamics, a butterfly that visits 100 plants is really a super spreader. Okay. And some of you who are home gardeners may have run into some of these ear rots or corn smuts. They're actually multiple types of filamentous fungi. They grow on and through the silks of corn plants, and they infect the seeds. And then the next year, the plants or the plants that are produced from those seeds will carry that fungus and produce spores. Okay, that's just a couple of examples. There are literally hundreds that I could talk about. And really, this talk will focus on a couple of serendipitous findings that we made. And a few years ago, graduate student Elaine Valdivia was working on the role of an expansin protein in the pollen of grass plants, and she was working with corn. And let me tell you a little bit about expansins. They're cell wall loosening proteins, first identified in cucumber seedlings and later from other plant tissues. And what they do is plants have cell walls rather than just membrane. So this loosens the cell wall and allows it to expand, allows it to bend, do things of that sort. Okay, so it weakens the linkage between the cellulose microfibers, allowing extension of the cell wall. It's involved, expansins are involved in many aspects of plant growth and development, and the leader in this area is Dan Cosgrove in the biology department. Okay, expansins are also a superfamily of cell wall loosening proteins, and there are these called the expansin B proteins, and there's some that are made that are produced in vegetative tissue, controlled development in various ways. But there's one really interesting one that is a group 1 pollen allergen. Up to 10% of all the protein in pollen grains, and there's 23,000 different types of proteins in pollen grains. Mom doesn't make it easy to get from that stigmatic surface down to her ovules. But up to 10% of the protein in grass pollen is this one group 1 pollen allergen. Causes hay fever and seasonal asthma in 400 million people, about 20% of the people in North America, and it's produced by the pollen grain. Okay, so this is, well, what Aline did is she got these mutants who had a mu transposon insertion into the expansin gene that knocked out its performance. Okay. And created 3 types of plants. We have the wild type expansin, we had the heterozygous plants, and then we had the homozygous mu insertion plants. Okay. And Aline was interested in assessing pollen viability and in vitro pollen performance from each type of plant. So when she looked at viability, she found there was no difference in the viability of the pollen produced from those 3 types of plants. In addition, she found there was no significant difference in average pollen tube length in vitro. So then she assessed in vivo pollen performance of the wild type and the double mutant plants. And what she did is she made controlled pollinations, and then she counted the number of pollen tubes 10 centimeters down the silk at 8 hours after pollination. She also looked at pollen tubes at the ovules, all the way at the bottom of the silk after 24 hours. And what she found is that at 10 centimeters down, she found that there was about 2.5 pollen tubes per silk after 8 hours, but less than 1 pollen tube when she made the pollinations with the double mutant. So it seems like that the wild type, the expansin, is playing a role in the pollen tube growth rate. Okay. Well, 24 hours after pollination, she found that the wild type had fertilized 39% of the ovules, but none following pollinations with the double mutant. 30 hours after pollination, 95 or 97.5% of the ovules that had been pollinated, whose silks had been pollinated with the wild type, had been fertilized. Only 20% of the mutants. But by 48 hours, 4 days later, basically all of the ovules had been fertilized by both types of pollen. Okay. Well, interestingly, she was growing lots of these wild type plants in the field. Okay? And then she was pollinating them with the different types of the three types, the wild type, double mutant, and the heterozygote. But 2003 was the wettest summer in recorded history in central Pennsylvania. It was cool and damp all summer. And what she found is that on the plants that she pollinated with the wild type, about 25% of those plants got ear rot, had this fungal disease. But if she pollinated them with the heterozygote, about 70% of the ears had ear rot disease. She used the double mutant, 72% of the ones she pollinated that way had ear rot. And we asked the question, so this just happened naturally in the field, she's going crazy, and I said, let's count it, let's study it, let's look at it, something interesting is going on here. And we asked the question, does slow pollen tube growth increase exposure or incidence of fungal sexually transmitted diseases? So she did, one of the experiments she did, I'm only going to talk about a couple, she took the silks, made them all the same length by cutting them off, and on one side she put the wild type pollen, and then on some of the other ones she put the wild type, some she put the double mutant, some she put no pollen. Or she put no pollen over here and then varied the wild type, double mutant, and no pollen. Well, what she found is that as soon as the pollen tube entered into the ovule, and I mean as soon as it entered in, the silks collapsed. There was an ethylene burst, and the silks collapsed. Not only did the silks collapse, but within a few minutes after the pollen tube entered, you could already see the formation of an abscission layer. These silks were going to fall off. So it was blocking that entrance. Okay, and so if you look at silk collapse, you know, it's greatest if you're using the wild type pollen, and least if you're using the double mutant. But there was something interesting that continued to go on too. When she did the half and half experiment, she found that the silks, until there was fertilization occur, until the pollen tube had entered the ovule, those silks continued to grow. So she was finding that those silks were 4 centimeters longer after she cut them off. So these silks continued to grow. So they had larger surface area from which pollen could land, okay? And in fact, there was no difference in the silk growth between the double mutant and no pollen in terms of total silk growth, but significantly less silk growth. So exposure rate to the fungal diseases probably went up with the slower growing pollen. So silk abscission would limit the exposure to smuts and rots. And slow pollen tube growth would also increase the exposure to smuts and rots in maize. So you can look at this silk collapse, this rapid abscission of the silk, as really a way of limiting the transmission of fungal diseases through the sexual organs in these plants. Okay, now a different serendipitous story. Here we have another plant that I worked on a lot over my life, is Cucurbita pepo subspecies texana. It's a monoecious vine with indeterminate growth and reproduction. That means it grows a little bit, produces a flower, grows a little bit more, produces a flower, so on and so forth. Monoecious means that it has separate male and female flowers on the same plant. The plant is a hermaphrodite, but it has separate flowers. These flowers last for only one morning. They open around sunrise and they close up around 10:30, 11 in the morning. Okay. We were at the time looking at the effects of inbreeding on resistance to herbivores and the diseases vectored by the herbivores in this wild squash. So that's what we were really interested in. And one of the herbivores are cucumber beetles, and here's the striped cucumber beetles. Spotted cucumber beetles are also common in central Pennsylvania. These are specialists on the Cucurbitaceae. If you've ever had a garden, you've seen them, or grew squash. And feeding in these plants, you can't get them to eat anything unless you smear cucurbitacins on it. Then you can get them to eat filter paper, whatever you want. If you put cucurbitacins on it, they love it. However, it's one of the most bitter compounds known to humans. Humans can taste in just a few parts per million of cucurbitacins. Okay. But these cucumber beetles also vector Erwinia trachyephylla, which is the causative agent of bacterial wilt disease. And at the time we started, what was known is that these beetles, when they're chewing on the leaves, their fecal pellets fall onto the open wounds, and the bacteria gets into the plant via the feeding on the open wounds. And once they get into the plant, they proliferate in the xylem. It's actually Erwinia trachea loving. So it proliferates in the xylem of the plant. And in fact, if you make a clean cut through the stem and pull it apart, because these Erwinia secrete an exopolysaccharide, a sort of mucilaginous snot-like material, you can pull it apart and you can see exactly where the plant plumbing system is located in these plants. Okay? And that's all exopolysaccharide and bacteria in here. The first symptoms you see is one of the branches will collapse. Looks like it's wilting. Even if you water the plant, it'll still look like it's wilting because their plumbing system is all clogged up with this mucilaginous material. Okay. And then after about 10 days or 2 weeks, the plant's completely dead. So it's highly fatal. The symptoms are visible. Field diagnosis is rather easy. Okay. And in 2002, 2003, 2004, we were looking, the inbred plants in the field had more beetle damage than the outbred plants. Wow, I'm thinking. Therefore, the inbred plants are having greater exposure to the beetles transmitting wilt disease. Okay. And perhaps, and I thought, well, also inbreeding tends to reduce resistance in general. Perhaps inbreeding also makes it less resistant to the exposure that it's getting. So greater exposure, less resistant. Being nobody's fool, I then designed some experiments. We were looking at the effect of inbreeding on individual incidence of wilt disease, and we predicted that the inbred plants would have a higher incidence than the outbred from cross-pollinated, the progeny from cross-pollinations. we also wanted to look at the effect of genetic heterogeneity at the population scale on the incidence of wilt disease. And we predicted that fields that had only inbred plants would have more wilt disease than mixed fields that had both inbred and outbred, as well as inbred and outbred plants from multiple families. So, planted 4 1-acre fields, 180 plants per acre. In 2 of the fields, all of the offspring were inbred, or the result of self-pollinations. One all had the progeny from one family, and the other field had only had progeny from a different family. Then in our heterogeneous treatment, we had 2 fields. There was a mix of self and outcrossed offspring from 5 families. So we had 2 inbred only, 2 genetically diverse populations of plants. Okay. Well, we found that the self plants, just like we'd hypothesized, self plants had more beetle damage than the outcross plants in the mixed fields. Okay? So the beetles prefer to feed on the inbred plants. Okay. They're less resistant to the herbivores. However, we found more wilt disease in the mixed fields than in the self-only fields. So here's the incidence of wilt disease in the mixed fields. Here's the 2 fields with only inbred plants. Well, it was even worse than what we expected, because when we looked at the inbred and outbred plants in the same field, we found that the outbred plants, the outcross plants, had a far higher incidence of wilt disease than the inbred plants. Well, I blamed all of this on Matt Ferrari because he helped me design these experiments. Okay. But Matt says it's probably a spatial artifact. And he plugged in all the data from our field and where each plant was located in the field and that sort of thing. And he played with different models and so on of spread and so on and so forth. Nah, it wasn't a spatial artifact. Jim Windsor, my collaborator from the Altoona campus, said it's differential susceptibility to Erwinia. So he figured out the LD50 and was inoculating the stems of these plants and so on and so forth. And he comes back and he says, no, it's not differential susceptibility to Erwinia. So what's that lead? Differential exposure to Erwinia. But we already know that the cucumber beetles are feeding more heavily on the inbred plants than the outbred. But when we looked at the data closely, we find prior to flowering, more wilt disease in the inbred only fields than in the mixed fields. And in the mixed fields, there was more wilt disease on the inbred plants during the month of June. Okay? Once they started to flower in late June, early July, then everything switched around. Well, we were not measuring anything to do with the flowers. Remember, these are no deposit, no return flowers. They open for a few hours in one morning and then are thrown away the next day. Okay, why would you care whether they're chewed on or beetles are there or anything? Okay. So, started working on the chemical ecology of cucurbita. We looked at the blossom volatile, at the cucurbitacins, blossom volatiles. Several compounds that are highly attractive to the beetles are known to be produced in the floral bouquet. Okay. We also knew from being out there and watching these plants that in the mid-morning, the beetles aggregate in the flowers to mate and feed. Well, most plants, most flowers don't have this many beetles. Of course, you take a picture of the most dramatic ones. But But what happens, it's a lek-like mating system. The males come in and hang out in these flowers. Females come in and choose a particular male. We don't know what the mechanism of the choice is. I'm guessing it's whichever male has gathered the most cucurbitacins, because cucurbitacins are also in the seminal fluid and passed on, and the females use them to wrap around the eggs and protect their eggs. But I don't see how these beetles expect to attract a female when they're defecating right into the place there. Okay. So they defecate into the flowers, and in some cases right onto the nectaries. Beetles will also do some chewing on the petals, anthers, and pistils. But this could explain the difference in exposure if it can be sexually transmitted, this disease, because the outbred plants produce more flowers than the inbred plants. Moreover, the outbred plants attract more beetles per flower than the inbred plants. They smell louder. That's also Matt's word. Okay, so here's an example of a male flower. Here's the nectary down at the bottom. There's 3 of these slits, and they're pretty big. And that's a microcapillary tube that we can insert in there and pull out the nectar if we want. Here's a female flower. The female flowers also have, the nectaries are more open at the bottom. So what we did is isolated Erwinia bacteria from field-grown plants with wilt disease. We grew the bacteria in culture, diluted the culture, placed a small amount of the culture onto the nectaries using microcapillary tubes. And we also did controls. Okay, but first we removed the nectar from these flowers, just like a bee would. Well, what we found is that, and we did it on female flowers that we pollinated and female flowers that we hadn't pollinated, but what we found in both cases, that we got over 50% of the ones where we put the Erwinia right onto the nectary, just squirted it right on. Here you can see Maruna sasu just squirting a little bit of this bacteria broth onto there. Well, they developed the disease. Male flowers, about 24% developed the disease. When we did stem inoculations, that is, injected it right into the plumbing system, 100% of those died. And if we use just broth and stem inoculations, nothing died in the greenhouse. Okay. And there's the very first plant that got sick. And so we're thinking it can be transmitted through the nectaries. So what we did next is we transformed Erwinia with green fluorescent protein. And here it is growing in our media, GFP Erwinia with green fluorescent protein. Then we took some of that and put it onto the nectaries of flowers. And 24 hours later, we were finding it at 5 millimeters below the nectary. Okay. And here it is glowing in our picture. That's Erwinia. So we collected male and female flowers in the late morning from the field and on a couple of dates in August of 2008. We then took these flowers in and we collected the frass from these flowers. We subjected the frass to real-time PCR with Erwinia-specific primers to get an idea of the exposure rate in flowers. Well, it turns out that 52% of the flowers in August had frass actually on the nectaries. 97% of the flowers had frass that amplified Erwinia. Now this is extremely high exposure rate because remember that the outbred plants were producing more flowers than the inbred plants, but by August, these plants are producing 15, 20, 25 flowers per day. Okay, they grow, each branch is making flowers, that sort of thing. So then the question became, well, the exposure rate is so high, how do you explain the fact that you don't have all of your plants dead in no time? Okay? What's limiting the transmission of this disease? Well, here we went back to the children's stories, and I asked the question, why can Winnie the Pooh store his honey at room temperature even after he puts his dirty paws into the honey pot? I'm asking you. Haven't you ever thought about these things? Well, the reason Winnie the Pooh can do this is that the nectar of many species contains plant secondary chemicals, some of which are known, some of which are thought to be have antimicrobial properties. And in fact, an old-time remedy when you have a sore throat or something of that sort is to take honey and a little bit of alcohol and use that as a cough soothe the throat. Well, it makes good sense because honey does have antimicrobial properties in it, which are derived mostly from the floral nectar that they've been consuming. Okay. At the time, that adaptation was thought to have evolved so that nectar doesn't ferment. You know, of course, if you have a sugar solution sitting out in the sun, it's going to turn to alcohol pretty fast. So that was, everybody just assumed that it was anti-fermentation. We asked, could it have also have evolved to prevent sexually transmitted diseases? And does the nectar of cucurbita have antibiotic properties? And what are the properties effects on Erwinia? So we did these diffusion assays using nectar from male flowers on plants that were heavily damaged by cucumber beetles and or lightly damaged by cucumber beetles. And the reason we used 2 damage levels, and this was done with field plants, is because cucurbitacins in some of the plant parts are known to also be induced by herbivore damage. It's produced constitutively, but there could be extra production. And we used ampicillin or 40% glucose as a positive and negative controls. The sugar concentration in cucurbita nectar is about 40% glucose. Okay. We used 2 strains of Erwinia and E. coli in this study. And what we did is we grew a lawn of bacteria, either E. coli or Erwinia, onto a petri dish. Then we cut out a filter paper disc and put it there, and then on the filter paper disc we either put ampicillin, glucose, or nectar from a damaged collected from a flower on a heavily damaged plant, or one collected on a lightly damaged plant. And we measured how much area, the inhibition zone, how much was cleared around that. Okay, so when we used E. coli, what we found is that there was a little bit of an effect. It's significant, but not highly. When we use the nectar compared to the glucose, but everything, the ampicillin just cleared out the E. coli, big areas. But when we grew Erwinia on these petri dishes, what we found is that at 8 and 12 hours, the area cleared by the ampicillin was not much different. It was actually statistically the same as that cleared by the nectar. So the nectar was as good as ampicillin at slowing or killing and preventing the growth of Erwinia on Petri dishes. By 24 hours later, the ampicillin was far superior. Okay, so then we can ask the question, is 12 to 24 hours of inhibition sufficient to deter Erwinia? Well, here you go back. Here's a flower bud, a male flower bud, and I can tell from having looked at these for many, many years, this is a male flower bud that will open tomorrow. Okay? This is a male flower that was open yesterday. If you just touch that male flower, it will fall off. And if you look at what happens after it falls off, you don't have to pull it or anything else. If there's wind, it's already on the ground, if it's a windy day. You don't, but if you look here, there's a complete excision layer. That's a wound that has healed completely. Okay, so they upsize these flowers. So by 24 hours, the flowers can fall off. But probably the abscission layer was formed at some time prior to that. Okay. So we went back to the greenhouse and did these inoculation experiments again. When we got about 50% of the flowers. We inoculate 50 flowers, 24 later developed symptoms, 24 of the plants, so about half. But when we left the nectar in the flowers, we only got about 12% of the flowers transmitted the Erwinia. Mock inoculations gave nothing. Stem inoculations gave 100%. Okay. So the combination of antimicrobial nectar and short-lived flowers, open for 6 hours, completely upsized by 24 hours, provide resistance to a deadly pathogen that can be transmitted via the nectaries within the flowers. So remember, virtually all of these plants are getting exposed in August. In our fields, we're getting exposed to Erwinia from multiple flowers on each plant on a daily basis, but yet only a couple developed symptoms per week. And this is the work primarily of Marunas Salcid. Okay, now, can we ask, are there other ways that prudent plants practice safe sex? Well, one is the floral volatiles. They serve to attract pollinators to the plants. Okay, pollinators are attracted with the floral volatiles. So everybody assumes that these plants, that these floral volatiles evolved only to attract pollinators to the flowers. And here's some of Matt's work as a graduate student. He looked at the volatile profiles from inbred and outbred cucurbita plants, and he found that each of those peaks is a separate compound, and the area under the peak is proportional to the concentration. And what he found is that the inbred plants produce fewer floral volatiles than the outbred plants. This probably explains why the cucumber beetles, there were more cucumber beetles in the outbred plants than in the inbred ones. But at the same time, many floral volatiles are also now known to have antimicrobial properties. When these floral volatiles are genetically downregulated, the incidence of diseases transmitted through the stigma and style increase. So what I actually think is happening is that these floral volatiles are fumigating the area, the stigma, the style. They're fumigating the bees that come into the flowers. And so, but bees have learned to associate these fumigants with rewards. So the whole bee visitation and pollen collection and things of that sort is probably evolved after the production of the volatiles. They were there to prevent sexually transmitted diseases in wind-pollinated plants. Okay, most plants have diseases that are transmitted through their sexual organs, especially via nectaries and the stigma style, and into the plant as a whole or into the seeds. So it's a great place to infect the plant because you can get your, you can be transmitted to the next generation vertically. Floral features that have historically been interpreted in the context of pollination may also play a role in the prevention of diseases transmitted via the floral organs, and floral traits that have been implicated in the practice of prudent sex in plants include antimicrobial nectar, continuous nectar production versus single pulse nectar production, where you put in a lot of nectar once and then don't put in any more. You have continuous nectar production in some plants. Antimicrobial floral odors, short floral duration, rapid abscission of the style after the pollen tubes have entered the ovary. And in fact, I wrote a little piece at the beginning of the New Phytologist when the first of the floral volatile papers came out, showing that the floral volatiles have antimicrobial properties and function in the in fumigating the flower. Well, anyway, if you go back and you look at if you look at this, if you compare dioecious plants where you have male flowers and female, the males last longer than the females, things of that sort. The males take more risk in terms of disease than the females. Interesting. Okay, so when you look at a flower, floral design, or look at the floral display, instead of just saying these things have evolved to get the pollen out and to collect pollen, actually they may also, the ability to avoid sexually transmitted diseases probably also has influenced the design and the floral display. Thank you very much. I should mention, these are my faculty collaborators at the top. Matt was involved in much of this. Jim Windsor was involved in much of this. Cosgrove with the maize work. Mark and Consuelo in the entomology department were involved with the floral volatiles. A couple of postdoc. Well, Ermengarde wasn't really my postdoc. I borrowed her from Jim Tomlinson. Wow, it was her technician. Graduate students, Nelson Hayes, Maruna, Elaine, and then two from entomology, Carrie Mock and Lori Shapiro, and a bunch of undergrads. These are just the undergrads who have co-authored papers as part of this transect through my work. Thank you very much. Questions? Yeah.
How come the woolly is not resistant?
How come it's not resistant to the? I don't know. It could be the Irwini infects many different species in the Cucurbitaceae. So maybe there's variability from one species to the next.
So that's obviously strong and very interesting hypothesis.
Yeah. And also, I think for most things, that this exopolysaccharide plays a key role in preventing the uptake of the toxic compounds and things of that sort. But that's a good question. Yeah.
I was struck by how powerful the antimicrobial activity was of the nectar, and meeting that with your translational work in science. Is there any interest in trying to discover new antibiotics which seem to be just as effective as our commercial antibiotic? Is there any rule of looking at places like this that we wouldn't normally think of for new drugs?
Well, I do know that there's a group at UMass Amherst that immediately started looking for what the chemical composition was and what the antimicrobial compound could be. And I also know that there's a lot of people looking at natural plant products for antimicrobial compounds and things of that sort. Not necessarily in nectar.
I don't think people thought of that.
Yeah, I don't think so either. Yeah.
On the other end of the spectrum, besides plants that jettison their flowers very, very quickly, there are these other plants that hold flowers for such a long time, weeks at a time.
Yeah, orchids.
Yeah. Have they particularly been examined for their ability to resist these microbial infections?
No, they haven't been examined. In fact, because orchids are not that, they're not used as a laboratory animal very often at all. And we don't know much about what diseases they harbor, how they're transmitted, or anything of that sort. Most of my career I've worked with a wild tomato that where the diseases of tomato and some of the genetics of tomato and that sort of could be imported, or I've worked with wild squash where I could, people know about the diseases of squash and some of its chemistry and things of that sort. But for the vast majority of plants, we don't have any idea what their diseases are, what herbivores eat them, that sort of thing.
So the male flowers, you said they fall off after about 24 hours. Presumably,
Yeah.
the female flowers must stay on or something. I don't know plant biology that well.
Yeah.
But they must still be a
That's exactly, the petals fall off, the stigma is upsized, but of course the ovary has to continue to grow.
And so are there other mechanisms in place to try to get extra disease protection in the female flowers, like a higher concentration of volatiles or anything like that?
One of the things, if you notice, the female flowers are oriented horizontally. They're out at the tip of the branch, and they're oriented horizontally. Consequently, not as many beetles aggregate in there because they're not as visible. The beetles tend to aggregate into the upright male flowers because they're there as part of a lek-like mating system. And I think also if you have beetles in the female flowers, any fecal matter that is falling down, not falling onto the nectaries as much. So I think that may have played a role in how the flowers are oriented and things of that sort. But, yeah.
The vision next time I go into florists, that the whole thing is just about fumigation.
Yeah.
It seems really interesting to me. I imagine in a floral shop there is been some selection for plants that humans like the smell of. So that might actually reduce the fumigation success. But that made me also wonder if you could say that the compounds that are involved in these volatiles that come up the flowers are more fumigant than you would expect by chance of possible volatiles. So it's not just any old compounds that are coming off. These are big compounds selected for their fumigation effects. So there must be all sorts of volatiles that plants could have been producing, but they've been selected to produce especially effective fumigants.
Well, I'm under it's my speculation, and this is just a gross speculation, the volatiles have evolved primarily because of their antimicrobial compound. for example, the question has always been, why do plants produce these volatiles? There's complex chemical pathways. When you walk through a field or something of that sort and you're smelling like pine trees or something of that sort, why would they make those when their herbivores are tuning in to these volatiles to find the plants? Herbivores navigate in an odor world. That's how they find their plants. Well, I'm thinking that these volatiles are coming out through the stomates on the undersides of the leaves, and these are holes right into the plant where gas exchange occurs. And what I'm thinking is that these fumigants are coming out of there all the time just to prevent fungal hyphae, bacteria, things like that from entering in through the stomates. When a plant is chewed on by an insect, it regulates additional volatile compounds. And, it not only makes more volatile compounds, but it makes a greater diversity of volatile compounds. And then I think that the original role of that was to fumigate these open wounds on the leaf surface. Having said that, secondarily, much like in my world, pollination, secondarily, predaceous insects have cued into these damaged volatiles, and they come by. So if a plant smells like it's been chewed on by an insect, then all of a sudden parasitoids go to it, other types of predaceous insects and that sort of thing. But I think that the volatiles were there first, as an antimicrobial function. But that's 97.5% speculation. Yeah.
So Andy, last question. After 40 years in science, can you offer some advice to the young trainees in the room? So PhD students, postdocs, what would you say to them? It's an unfair question. I'm putting you on the spot.
Yeah. Okay. My advice would be, don't play politics. Have things that you want to do and pursue them with great vigor. You know, have ideas that you want to test out and pursue it with great vigor. It's actually an honor to be a faculty member in a way, because the only thing, mostly, to be able to do what I want to do in terms of research is, if you work for a company, you may get paid a little more money or something, but they're telling you what you need to do. And if they say, that's not interesting, then move on to some other project. They decide these things. But I get to play with whatever I want to play with in my lab, whatever's interesting me at the time. And that's a tremendous honor and an opportunity that not many people in the world have. So have ideas, pursue them with vigor.
Thanks so much, Mark.